Hello students, welcome back to the channel. In this video, we are going to study about enzyme inhibition. See, it is inhibited by many non-substrate chemical agent. Now see, enzyme inhibition can be reversible or it can be reversible. So see here right now, this is the example of irreversible inhibition. So in irreversible inhibition, what will happen? Inhibitor will form strong bonds, which is unbreakable. Means the enzyme is now unable to bind with any substrate and reaction cannot move. Whatever extra substrate is present, but reaction can never occur in case of irreversible inhibition. Is it clear? This is irreversible inhibition in which once the inhibitor binds, now substrate cannot bind whatever number of substrate you increase. Next is reversible inhibitor. In reversible inhibitor, main thing is this, that the inhibitor is making weak bond. Okay. Inhibitor is making weak bond with the active site. And the effect of inhibitor is not permanent. The bond can be broken and substrate can enter inside. And you can say, though inhibitor will slow down the reaction, but it will not stop because substrate has capability to bind. Now next is reversible inhibitor can be of two types, competitive or non-competitive. We are classifying them based on where the inhibitor will bind. See here in this diagram, this is a substrate. Clear? This is substrate and this is inhibitor. Before substrate can bind to the active site, inhibitor is binding to the active site. This type of inhibition is competitive inhibition. In this case, the inhibitor is actually resembling structurally to the substrate, fit into the active site, and as a result, enzyme cannot do the work. But main thing is this attachment is only temporary. If you increase the substrate concentration, the inhibitor will not be able to bind to the active site, and active site will be occupied by the substrate. So in case of competitive inhibition, substrate, which is inhibitor, is resembling the substrate and inhibitor is binding to the active site before substrate can bind. Next is non-competitive inhibition. Understand the diagram? See, this is enzyme. An inhibitor is binding to this site. This site is known as allosteric site. And such type of enzymes which are having two sites, active site for substrate and allosteric site for regulator molecules, these are known as allosteric enzyme. Here it is an inhibitor which is binding it, and as a result, see beta active site shape is changed and now substrate cannot bind. Means in this case, non-competitive inhibitor, the inhibitor is not binding to the active site. It is binding to the site other than the active site. Is it clear? So this is your non-competitive inhibition. So I think so two type of inhibition is clear. Competitive, where the inhibitor is binding to the active site before substrate can bind. And in case of non-competitive, inhibitor will bind to the allosteric site and will change the shape of the active site. Next is irreversible inhibition, in which the permanently destroy of the enzyme is done if the inhibitor bind to it. Example, poisons. Many poisons are irreversible enzyme inhibitor, like mercury and lead. They bind irreversibly to denature the proteins. Certain nerve gas, they poison the enzyme acetylcholine esterase, this enzyme, which is important for the function of nerves and muscles. So these are the few examples where the inhibition is irreversible. You cannot reverse it. Now enzyme is permanently damaged. Another is cytochrome oxidase, which is used in the electron transport system. It is sensitive to cyanide. And cyanide, what is the problem it is going? This enzyme is irreversibly inhibited and cannot transfer electron from its substrate to oxygen. So I think so up till here we understood reversible inhibition, irreversible inhibition. Reversible inhibition is of two types, competitive and non-competitive. Next, we are going to discuss about feedback inhibition. In case of metabolic pathway, if one product of one step is used as a substrate in the second step and continuous chain reaction is there, okay, 4-5 steps are there and the chain reaction is there, then what happened? The end product, if it is in excess produced, if the end product is produced in an excess, it will bind to the allosteric side and will stop the first step only. So different enzymes you are using it to catalyze each step, and the final product may inhibit the activity of first enzyme. So this type of enzyme regulation in which the product inhibit the first step only is called feedback inhibition. So I hope so this video might be helpful for you to understand what are the different type of inhibitions which are working in the enzyme. Please understand the concept properly, and if you like, then please subscribe to my channel for more videos. Thanks for watching.